I'm Johnathan Mack, and I'm currently a student at Carleton University, and I focus mostly on microfungi, which are, as the name may imply, microscopic fungi. They are found pretty much everywhere and pretty much unknown for most people around there. So I want to introduce some of my favorite species and a brief introduction to a special group of them, which are called Iphomycetes, which I will discuss about further in a few minutes. So I have to introduce Iphomycetes as asexual fungi. Most fungi that you find in the forest, like the big mushrooms or the chanterelles or the morels or whatever, are sexual fungi, which raise from the union of two parents, just like you and me do. However, asexual fungi are a bit different in the fact that they reproduce more or less clonally, and that they do not require two parents to occur. It would be a bit like if your fingers, for instance, just decide to fall off and decide to become new you, which would be identical as you, but without the help of any other individual. Those asexual states can be found with the sexual state, and which would be the moral or whatever in that case, and the sexual state would be called the teleomorph in that case, and the asexual state is the anamorph. Most of the Iphomycetes, which are a group of anamorphic or fungi, are microscopic and require the help of a microscope to see properly. However, some of them are quite big and can be found even with the naked eye in some cases, and such as those Elicosporium virescent on the upper right, which produce green patch on bark, or Gibellulas and Penicillium glandicola, which produce large stalks on their own specific substrates, which I will discuss a bit more in a few minutes. Don't worry, I won't forget about them. They are extremely variable, as you can see from those four species, and can be found virtually on any organic substrates. Some Iphomycetes are even specialized on extreme habitat, which include foodstuffs such as chocolate. I think one of the species grow in kerosene in airplane oil tanks, and some have been found growing in the walls of the Chernobyl nuclear reactor, where they feed on radiation. Iphomycete is not necessarily a real taxonomic group. It more or less denote as states of a fungi, and many mushroom and fungi have evolved the Iphomycetes state independently and all over the mushroom kingdom. However, in general, but I broadly in general, I just say, as Iphomycetes are usually the asexual states of cup fungi, such as Pezisa on the upper right, which should look like a little cup that you can find on oil, or a cup that look a bit like the Chlorosiboria, which are blue and grow on woods. Another group of asexual fungi that can also be produced by those cups, which the cups are telomorphic or sexual states, are Coelomycetes, which are very similar to Iphomycetes, but produce a more complex rocky structure around them, or stromatic tissues that we call. But I won't really discuss about Coelomycetes, at least not today. Unlike their telomorphic state, which reproduce with Basidiospores, which are formed on Basidia on gill mushrooms, or Ascospore, which are formed in a funny-looking sac in Ascomycetes. Iphomycetes produce their candida usually quite naked and without any of those structures that are always only found in the sexual state. I'll have to introduce one concept that is a bit annoying for people that are interested in mycology like me, and it is the one name, one fungus. Usually, the anamorphic state of the telomorphic state of a fungus was named differently. For example, on the upper two image are possibly the same species. On the upper right, it's the Trichoderma, the Hypochria state, which is the sexual state found on wood forming yellow cushion. On the upper left, it's the Trichoderma state, which produce a little blue puff on wood, which is quite common. Basically, you can almost see this as as one of the state being the caterpillar and the other the butterfly, pretty much, honestly. Because sometimes they are not found in the same habitat like butterflies and caterpillars. For instance, you may find butterfly and caterpillars on completely different habitat, one on leaves and another on flowers, just like Iphomycetes are in a different habitat from their sexual states. They had different names, but now this is not allowed anymore. And usually the oldest name or the more familiar name, which is usually the telomorphic name, is the name chosen for those organisms. Iphomycetes are unfortunately quite tricky to identify, and some of them unfortunately won't be identifiable without some lab work. Although fortunately for us, some of them you can recognize right away just by looking at them. One of those distinctive species would be the Armameys Orientation that you can find growing on rotten wood. It looked like a orange wet jelly, gelatinous, and it is pretty large too. It can reach a few centimeters in length, and it is quite conspicuous. However, some need microscopies and even molecular or cultural analysis, which I will briefly touch in a few minutes, but not for very long, as I want to focus more on species that are pretty and more distinctive that anyone here could recognize. Perhaps. They're tricky, even me, I struggle once in a while. So microscopy is quite important in those groups. There will be a lot of picture like this one that I've taken under the microscope. In this case, I think that this wall structure would be maybe 50 micron in length or so, maybe a bit more. For a sense of scale, I was curious this week and I measured some of my hairs, and the width of my hair, each individual strands were about 100 micron. So the length of this structure would be 2 times as thin as the width of my hairs, which show how small they are. Fortunately, compared to telomorphic fungi, they tend to be relatively simple, but unfortunately, biologists really like to name everything they see with big, fancy, and odd names sometimes. So the names for all of the structures that are used to identify those mushrooms are quite unique, and probably most of you are not familiar with them, unfortunately. So I suppose that most are aware that the mushroom reproduced by spores. In this case, the asexual spores, which work pretty much the same way as a spore, regular spores to be dispersed into a suitable habitat, is called the conidium. The conidium tend to be produced on a modified cell, which is called the Carnegiogenesis cells. And the Carnegiogenesis cells tend to be attached usually in groups on another specialized cell called the conidiophore. Personally, I think that especially the term Carnegiogenesis cells and conidiophores are sometimes a bit ambiguous, so I will use them somewhat interchangeably at time, even if I probably shouldn't. I'm sure that some might be a bit angry at me, but it's okay. And another part of the mushroom that most people are probably familiar with is the Misalyam, which is formed of long threads, as you can see on the bottom, with numerous cross walls inside, which are the darker line inside of the thread that you can see. These are normally the non-reproductive parts of fungi, and it is used mainly in feeding or perhaps reaching other habitats. I'll also use the term hyphae quite often. Usually, I use the term hyphae for a single strand of Misalyam. So I will use the term hyphae quite a bit later in this presentation. And hyphae is, as I just said, simply just a smaller part or a single strand of the Misalyam, as you would see on the top. A few of the species of Hyphomycetes are a bit unusual because the Misalyam itself break up in many pieces, which tend to be rectangulars. And those are act just like Canadia and are dispersed everywhere. This method of reproduction, which is quite unique, is called Tilak reproduction. And I don't really have any example of this today, but I just want to show everyone that this type of reproduction can occur in some species. So as I said before, the Carnegiophore is a specialized hyphae, which hold the Carnegiogenesis cells, which themselves hold the Canadia. The Carnegiophores can be extremely variable. As you can see on the Aspergillus on the upper right, you can see the little almost bowling pin shaped Carnegiogenesis cells, which are holding some round Canadia, and a big bulbous around the Carnegiophore. In some Hyphomycetes, however, it is not that distinct, and it is almost indistinguishable from the surrounding hyphae. I think probably the most important and the most variable structure in Hyphomycete is the spore or the Canidium itself. They can be of virtually all the shape imaginable, ranging from the single circular shaped Canidia from Penicillium to the extremely odd shaped Canidia from Lateri remulosa, which kind of look like almost look like a shark swimming right through you. And because of its unique shape, the Canidia of this species can be recognized by its shape alone. The Canidia can also be variable in color, and color is also a useful trait in the identification. Again, I will use, like many biologists, some fancy term for colors. Usually pale or transparent Canidia are usually called hyaline, while the Canidia, which are brown or black, are usually called Nottyakis. There's also other variation in colors. Some Canidia are even red or yellow, but those are less common than dark, almost black Canidia or just transparent Canidia. In some species, the Canidia are almost stuck together in chains, and they kind of almost look like trains from the tops, honestly, with all the wagons stuck together. The presence of chains is also very useful to identify a species. And in some species, those chains can be branched and to the point that the Canidias look like the former branch of a trees, which is well seen in this picture of the Armameys in the lower right. Canidia can be dry and powdery, which make a powdery mass on the substrate or where it was growing on, or they can be sticky. In that case, that the growth will appear as a jelly or more gelatinous. As I said before, Canidia are very variable in shape. There is fancy name also for Canidial shape, unfortunately. Like simple Canidia without any crosswall are called Amerocanidia. If your Canidia have a single crosswall, they are called the Didymocanidia, and they are said to be one septate, which is a term that I will use once in a while. And if your Canidia have a lot of horizontal crosswall, They're called Phragmocanidia, and usually I call them by the number of septum. In this case, there are 7 septums, or there are 7 septate Canidia. Some Canidia almost look like ear of corn with both vertical and horizontal cross wall, such as those Berkeley Asmium, which also show an example of a Dematiasis Canidia, which is quite dark. Those are called Dictyocanidia. Some Canidia are coiled on themselves, looking almost like sleeping rattlesnakes or something, and those are called Helicocanidia. Or if they're looking flat under the microscope, they're 2D Helicocanidia. But if they look 3-dimensional, like a bobber, they're called the 3D Helicocanidia. They also have arms arms sticking out, and those are called Sterocanidia. And an unusual subset, which look like Dictyocanidia, but even bigger, is the bulbils, which can be so big that the individual one can be seen without the aid of a microscope. The act of how Canidia is formed is called the Carnegiogenesis, and this is very important for identification of fungi too, although this often requires the help of a good microscope, so it might be hard to see. Many species like this Aspergillus have its Canidia formed almost like a balloon on top of the hyphae and just inflate and eventually get released. And this is called a phylitic Carnegiogenesis. Other species, such as the Basidiopycnides, produce some sort of odd scar each time a Canidia break, and then the Carnegiogenous cell grow up a notch, making a multiple ring under the Canidia on the Carnegiogenous cells. This is called the analytic reproduction, and I won't focus on this one at all because it is extremely hard to see the rings. Even if you have a state-of-the-art microscopes, they're an absolute pain to find, honestly. Some species, like Brachydesmiella biseptata on the lower left, break off from and leave some sort of scar-like looking structure on the conidiophore carnigenous cell, while species such as this Brachysporium form on almost little tooths, which are called denticles, at the top of a conidiogenous cells. One conidiogenous cells can have many denticles in some case. Since I talked a bit about the Phylites earlier, which is probably the most prominent Carnegiogenesis method. There is 2 main types of arrangement in Phylites. One of them, which is seen as Misalyam, is called the penicillate arrangement, where all the phytes raise up like that, like very tall and kind of look like ants almost. Another arrangement that can occur in other species, such as the Cladopotrium, is called the verticillate arrangement, in which the phylites are more lateral to the sides of the conidiophore. Another trait that can be found and is rather dynastic for Phylites is called the collarette, which is some sort of colors that occur exactly where the canidia where the canidia inflate. Unfortunately, most of the collarettes are not very conspicuous again, and those structures are often very hard to find even with a good microscope. In some species, this is taken somewhat to an extreme, such as Bloxamia truncata, in which the spores appear to form inside the carnegiogenous cell. There's other structure that you can find in some species, but most of them will lack these structure. One of them is the clamp connection. The clamp connection is found specifically in the basidiomycete, which include the gills mushrooms, jelly fungus, and rust fungi, amongst others. but not all species have them, and no cup fungi ever have clamp connection. So if you see this, at least you have a good lead to know that your fungi must be a basidiomycetes. And fortunately, most of the iphomycetes are not basidiomycetes, so this narrows it down. The clamp connection is simply a clamp that connected to hyphal cells at the crosswall or septum. Some species will have almost needle-like sterol hairs, which are probably self-explanatory, really, which are hairs surrounding the conidial mass usually. Other species have a mucilage, which is a sheet, almost transparent sheet that you barely can see, such as on these spores. The mucilage usually surrounds spores, and it is quite rare in iphomycete, and it is more common in ascomycetes, telomorphic states, and coelomycetes. And some conidia have a creepy appendage and look almost like monsters of some sorts. Those appendage can be hard to see, but are extremely helpful for identification. So it is worth to try to locate them because there are not many species that have appendages. Most species, unfortunately, are not visible to the naked eye, as I said before, but some are rather some are rather compact, such as Elicosporium, and some species are stalked too, which form a distinct group I will talk in 1 minute. Texture is also very useful. Into identification, like does your fungi look all gelatinous? Does it look all dryish? As those traits may help in identification. One subgroup of large iphomycetes that people around there can be on the lookout for and are spardochial iphomycetes, in which the conidiophore are grown in an extremely compact matter to form almost a cushion-like structure on the substrate. Usually I find those Spardochium on wood, and only certain species produce Spardochium. So the presence of a Spardochium is really useful for the identification. Most are quite delimited and really look like little cushions of some sort, and usually tend to be gelatinous, but some of them can be dry depending on the characteristic of the Canidia. Another subset of amazing iphomycetes that are worth to find around there, and that the specialist in this group isn't meeting at this very time is the Cinetophytes iphomycetes. They are similar to Spardochium in some way, but the Canadia are formed on a long stalk and in some species they tend to look like delicious lollipops, but they are way too small to eat. In fact, I don't really think iphomycetes can be eaten unless they're used to ferment food, but this is not something that I will discuss today, unfortunately. Cyanomatous iphomycetes, like Spardochium iphomycetes, can have their dry canidia, such as the Misalyam, which look all powdery and feathery almost, or have a dry or wet canidia, which make the Cinemata look more like a lollipop, such as this Trichoderma flavipes. They're really stunning and they are really a pleasant thing to find around. And I should note that all of those pictures, with the exception of the life cycle on the first slide, are my own pictures, and that all of those pictures have been taken into Ottawa area or at worst only a few hours away. So all of those species should be around if you know where to look for. So this is just showing the amazing diversity of cinematus fungi. The picture of Arthrosporium candidum in the center bottom is looking all blue because I used a special dye to look at the Canidia, which can be really helpful to see some of the finer details sometimes. However, unfortunately, most of the iphomycetes don't really fit neither of the previous categories and are called micronematous iphomycetes, in which the stalk is only a single hyphae. And as I've said before, they can be isolated or can be forming some sort of grouping like Helicomysis roseus. on the upper right, but not really true cushion like the Spardochium, and definitely not stocked with tons of hyphae like Cinematas do. So I'll discuss about a few of the habitat where you can find some species around, but realistically, any organic habitat will yield some hyphomycetes if you look good long enough. Some species, however, are rather specific to a single habitat, and others are broad generalists, which will grow on about anything. I will discuss about a few of the habitats right now. One of the most amazing and probably least known group or least expected group is the aquatic hyphomycetes. Usually, those species have highly modified canidia, usually with lots of weird arms, and because the canidia have such odd shapes, usually they are enough for identification alone. The best way to find those canidia is to look at some water foam under your microscope if you have one, or try to find a microscope if you can, because looking at water foam is really a lot of fun, and it not just for the canidia that might be lurking there. Water foam can be found near rivers or near any bodies of water and will yield a few interesting things for sure. Some IFO might be on soil, and but most of them are quite difficult to observe. However, some species produce a conspicuous fuzz like the Arthrosporium. I've also added the Costantinella, which is sometimes, often considered the anamorphic state of morels, which I suppose that many people around here are familiar with. However, even if you find a Costantinella, that does not mean that you will find morels, unfortunately. Or else I would probably find 1,000 of them into little forest near my house, which had Costantinella on almost all of their trees. Others, like the Artrosporium or the asexual state of Bethesda cup fungi that I showed earlier. Most I would say are found on wood, and I call them simply wood hyphomycetes. Usually, they're not really picky on which species of tree they will grow, but some are. However, they tend to be more picky on the state of the wood. Some prefer bark, some like all old rotten and wood, or some can be found on both. Wood is also well suited for damp chambering, in which I put a sample in a little box, which I keep it wet, and then I can look at under the dissecting microscope, which is a special microscope to look at very small thing on wood every few days to look at and maybe growing in the wet box. Some species will grow on leaves only, unlike the wood hyphomycete, and they are rather picky. In fact, some are very picky, growing only on one species of trees, such as many species of oidium, which are more well known as the powdery mildews that most people that work with plants would know. Another group that you may find on leaves are sooty molds, but these are less damaging, or at least directly, because they feed on secretion produced by aphids, which are also found on leaves. Some species grow only on other mushrooms, such as Cypadomium brunium, which grow on boletes, usually rotten boletes. Most species in that are not as picky as the leaf inhibiting hyphomycetes and prefer a specific subset of a fungi. For instance, the Blistum tomentosum, which produces beautiful white cinemas, grow only on certain species of slime molds, which are quite commonly found in the area. The odd-looking Tilak Lady brachiatum on the lower left grow on old mushrooms that grow on woods. Usually the smaller ones like mycenas, which I suppose that some people here might know about. Other species, such as the beautiful bright pink Eloquoid Christianseni, grow only on lichens, which is usually considered a mushroom as well. Amazingly, some will also grow on insects, and some are capable of killing their insects. People that may be familiar with fungi may be aware of cordyceps, which also grow on insects and kill them. And in fact, many of the fungi that grow on insects, iphomycetes that grow on insects, are actually the asexual state of cordyceps and their relatives. I particularly like the Isaria farinosa. Even if it's not really obvious that it grown an insect, you have to dig to the substrate to find the host, which usually is a moth cocoon. I think they should be called the fairy ice cream cones, though. Gibellula is also interesting as it grow on spiders, and the only clue that you have are the little legs sticking out. Not all, however, I just want to point out real quick that not all fungi that you find on insects are iphomycetes, especially as there is also Asperomyces eximidis, which is a bizarre but yet telomorphic fungi that produce small yellow stalks on living ladybugs. And this is one to look under the microscope if you ever get to see one. It's beautiful. Finally, unfortunately, a microscopy and habitat and all of those are not necessarily enough to identify fungi. And sometimes you have to grow them in a laboratory, like those which were grown in petri dishes with controlled food, controlled nutrient for them to stimulate their growth. In culture, they can be extremely variable, but more or less identical for the same species. So strains of the same species tend to be similar, and they tend to be unique. So there can be beautiful red ones, some that look fibrous, some that look gelatinous. Many will even produce their Canadia in petri dish, which make an excellent opportunity to try to look at the Carnegiogenesis of those Canadia, especially if you don't have a lot of specimen on your substrate before. Furthermore, those cultures should be sterile and therefore can be used to sequence your mushroom. Sequencing is basically just comparing the DNA sequence to other fungi, and closely related sequence will branch one to next other. In this case, the blue spot is an unidentified species from Montreal. It do not seem to be matching any of the known species in that group and may very well be a new species. In fact, there is a lot of new species that are yet to be known in the area in this group. So now that I've introduced a bit to the basics of iphomycetes, really the simplest basic, I want to introduce some of my favorite species that can be found in the area, although some are rather specific in their habitat. So the first species, I have about 30 species or group to introduce. So I will try to go rather fast and introduce them briefly and tell what make them interesting and what to grow on interseason if applicable. So the first one is Arthrosporium candida, which looked like almost a tree. And I just realized right now that I unfortunately forgot to say that the cinematus or the stalk, the iphomycetes, can be determinate if you can easily distinguish the head from the stalk, or indeterminate if you can't really see any head. Like in this case, it would be more considered a indeterminate cinemata because you can't really make up the head really. The Canadia are rather long for an iphomycete by measuring about 40 micron in length. In iphomycetes, Canadia tend to range anything about 2 micron in length to about 40 micron in length. There's a few exceptions that are much bigger and that I will talk very soon, though. It is somewhat rare under my experience, but I am pretty sure that it's because I'm not looking. It's easily overlooked, even if you look for them, because they grow on wet sticks late in autumn after the leaves fall out. So they're probably hidden by leaves. The telomorph of this fungi is unknown. It have never been sequenced, so we don't really know what its relatives are at this point. One group that I should introduce because it is quite ubiquitous and important is the Aspergillus. if you see SPP after the name, like in this case Aspergillus, is that's only mean that to describe all the species in that group. So, and I will just discuss broadly about Aspergillus in general because there's about probably more than 100 species around, at least worldwide, and probably several hundred yet to be found. So it's just too much to talk about. So the Carnegiogenesis cell is inflated and they look like little suns or flowers. And the conidiophore rather, and the Carnegiogenesis cell look like little bowling pins. The conidia are small and they usually form a dry chains that you can see a bit on the upper right, but those chains tend to fall apart when you do a microscope slide. They grow virtually anywhere and can cause infection in crops or even in human, where they can cause a deadly infection in rare cases. They also produce mycotoxins, so they're a group of ongoing research. On the completely different side is the Bactridium species, which are probably one of my favorite Hyphomycetes, if not my favorite, just because the giant Canadia, which reach about 250 micron in length and almost 40 micron in width, are simply all beautiful under the microscope and are very worth the effort to try to look at them. I'm pretty sure that there's at least 2 species involved in the area, perhaps more, but I've only found 2. The best way to distinguish between them is that the species 1 on the left have a Canidia with 4 cross walls inside of them, which means they are four-septate, while Bactridium species 2 have a Canidia with 2 or 3 cross walls inside of them. And this is rather constant, so one specimen will always have 4 septate Canidia, while the others will have 2 and 3 septate Canidia. I'm usually finding this species in late autumn on big but well-rotten logs where they form little cushion. They're rather uncommon, but I think this is because it is more of a fungi found in old growth forests, which are a bit rare in the area. Ovaria bassiana is probably one of the most common fungi that you will encounter on insects. And I usually find it parasiting and killing beetles and then overgrowing them all over almost like a snow. It is quite distinctive. Unfortunately, the microscopy is not necessarily necessary, at least to go to genus. But if you're inclined, you can try to look at the Carnegiogenesis cell that form little zigzag, but those are so small that they are very difficult to see. An interest of this group of fungi is that they also produce interesting toxins, and they can be used as biocontrol agent against insects and maybe nematodes, if I recall correctly. Chromalosporiums are one of our most common species. I suppose that many people will notice them after I tell to look for the white fuzz growing on sandy soil near trails and sandy forest, which is one of the species of Chromalosporium, usually considered Chromalosporium fulvum. There's other species that produce beautiful purple cushions like Chromalosporium corolescens, but this one is much rarer and I've only found it a few times. Unfortunately, do not stay purple for long as the Canidia are quite pale and it can turn a drab gray. The telomorphs of a Chromalosporium is usually considered to be a Pisiza, which is the common cup fungi. Another rather common fungi around there is, or at least Hyphomycete, is Crenula caliciformis, which produce enormous cinematas. At least for an Hyphomycete, it is enormous. But really, it's only a few millimeters tall, so this only shows how small those things really are. This species is rather common around here, and I tend to find it on large dead trees on the ground with bark still on, especially the bark with a bit of moss on it and cracks. It is quite common in autumn. While it is very distinctive into naked eye, under the microscope it is very difficult. All of the elements are extremely small. The Canadia are about 3 micron in length, which are about the smallest you can get for a Canadia. And Carnegiogenesis cells and Carnegiophores are all smaller and all packed together. Some fungi are just frustrating like that, unfortunately. Fortunately, it is pretty to look at, though. Cryptocarinium condensatum is an unique one, which I've only found once, despite someone told me that it is quite common when you know where to look for. Obviously, I don't at this point, but that's okay. I think the Canadia kind of look like little squids, and they do not really break up, so it's really a unique set of the Canadia type. They are dematiaceous, which means that they are darkly pigmented, and they are very, very tightly packed together, and they almost look like a crust on the wood, as you can see on the lower left. Dictyosporium is another one of those very fun fungi to find here. It is hard to believe, but this subset of Canadia shape actually have a name because biologists are really inclined to name everything, and they really need fancy names too. That's how we are. It's okay. So those Canadia are called a cheroid or cheroid Canadia, which I think literally mean and-shaped Canadia. And indeed, to kind of just look like ants saying hello like that, they're waving at you. The carnigenous cell in some species is inflated and can be attached to the Canadia as well, so that makes them quite distinctive, as on the upper, sorry. It forms very small Spardochium or the Canadia can be scattered apart. It is found on very wet wood. Endosporidoides pedicellata is a unique species in which the Canadia fragments. The Canadia also seem to have some sort of a sheet surrounding them. I'm not entirely sure if it is truly a gelatinous sheet or not. What makes this species so unique is that I'm probably the second person in the world to find it, which made me quite happy. The first person to found it about 15 years ago and named it from pogs in Hong Kong. So while the host and region can be useful for identification, they are not always limiting, and there are exceptions and surprise around. So even if your species look just like a species that was found in Hong Kong, you can't necessarily exclude it on this basis alone. I have pretty much no choice to talk about fusariums, even if I'm not sure if they're the most interesting fungi to look at. They're kind of cute. I won't say. And I know someone that might get a bit angry at me saying that, but it's okay. So some species like Fusarium merismoides, however, are quite easy to find and form a huge orange slime on wood, usually freshly cut wood, such as this one that was cut from a beaver at Mutt Lake, which some people around there may be familiar with. The Canidia are boat-shaped and often multiseptated. The main concern with this species, the Fusarium, is that some of the species are very notorious pests of plants and can cause devastating loss of crops, and especially in Western Canada. And therefore, they are probably the most widely researched fungi group in Canada, I would say. Raphiums are one of those cinematus fungi that you can find in various habitats. Only the stalk is pigmented or dematiaceous, and the Canidia are hyaline or transparent. It is very hard to see, but the carnegiogenesis of this species is actually analytic. And one of the clues for this is that the Canidia are truncated, or they almost look like there's an ending of them that have been cut. While truncated Canidia are very common of analytic fungi, they are not necessarily restricted to them, but it is a good clue. It is very common, especially on decaying litter, but some species such as Graphium curotium grow associated with insects, apparently, such as bark beetles. The aplotrichums are probably the fungi that the iphomycetes that I getting tired the most of because of how common and how ubiquitous it is. It is quite pretty, but it is rather plain. And since it is so common and so abundant, it does form it is a good species to try to learn your basics in iphomycete. Furthermore, all the structure like the conidiophore or the denticles, or which are from which the conidia are formed, are quite obvious, and even the conidia are quite large, reaching up to 15 microns in most species. So this is a really good species to learn the basics of iphomycetes, I would say. Also, it is super common and big and obvious. Its telemorphs look almost identical to it, but it have basidia, and it is a basidiomycetes that form crusts on wood, and it is really a distant relative of guild mushrooms. Although it do not form clamp connection like many basidiomycetes do. Probably the most striking group of Canadia is the Eliquid Canadia, which I've described briefly earlier as little snakes leaping and coiled together. And I really do think they do look like that. However, some species will uncoil in water. They are quite variable on wood. Some species can be pink, green, and pretty much any color in between. The Carnegiogenesis is rather variable and is a good trait in identification of those groups. And conidial size, width of a coil, and thing and coloration are all useful too. I usually find these species on very wet and rotten wood, and in those habitat it is very common, and you'll be bound to find one eventually. Another one that form Eliquoid Canadia is Helicune wicornis, which form unique 3D Eliquoid Canadia. Those Canadia are believed to act as bobbers, which entrap the air, which allow them to float at the surface of a water body. In fact, the forest where I find these specimens pretty much each year is periodically flooded during spring, but dry during the rest of the year where they can grow. During spring, I assume that the Canadia detach and just float all around the vernal pool, hoping to find another rotten tree to grow on, which I think is a very interesting mechanism. I don't know how common it is because I've only found it in that one forest, but in that forest it is very common. So it's probably picky in its habitat. I'm very delighted that all of my species with Eliquid Canadia started with H, Eliquoids, Helicune, and then Ialotrochophora lignicola, which is quite a mouthful, I would say. I mean, it is a really long name, but I think I've managed it. This one produced little, more or less indeterminate snemas, and the Canadia are a bit different too, as they do not form a complete coil like the other do. And the Canadia have three quite evident septums on them. And they do seem to have a unconspicuous gelatinous sheet surrounding them too. Although I don't see any mention of this in my books. It is common, but the cinemas are small and it is found on rotten wet wood, as again. Probably my favorite fungi because I worked on it for years trying to resolve what its relationship are, and I will try to finally publish it very soon, is Armameys Orientation, which produce gelatinous orange, very bright orange in fact, Spardochium onto substrate, which is usually rotten wood. The conidia are in long branched chain, which do not break easily, unlike most conidial chain, which will easily break up when covered with a cover slip from the microscope. It can be confused with several other mushrooms, surprisingly. It do resemble a jelly fungus, such as Tremellar Dacromyces, to which it is believed to be related by some, or even a slime mold, Plasmodium, which is the juvenile state of a slime mold. Leucoblue Egg Layer compressa is also an extremely common hyphomycetes. Probably one of the first one that you'd find if you're ever interested in looking at hyphomycetes in our local forest. Unfortunately, do not make a very good species to practice with for the microscopy because, well, it is in a sense because all of the elements are small, quite gelatinous, and tend to get stuck all together. But if you can manage, you'll see a beautiful verticillate pattern of phyllite like this, and not like this like most phylites, or at least the most typical seen phylites. Menisporod loci is also probably not a bad fungi to learn, and also a good model for starting microscopy, because if you're able to see its main diagnostic trait, you know that you have a good setting or whatever setup that you use to see it, because it is small but distinctive. Menisporod loci have canidia which produce little appendages on each side, which you can barely make out in my picture until lower left. But trust me, they are there. If you look close enough, you'll see those little needles poking out of the canidia, which is rather unique for this species. I think there are species as well without appendages around, but I've never seen them yet. The most common is the one with appendage and canidia with 3 septums. It is common on underbark of maple in autumn. Monodictis is a group of fungi, more or less a wastebasket, I would say, in which every canidia that kind of looked like the one on the upper right were called a monodictis if they were growing solitary and they were dry and do not really form any Spardochium. Although I think that some monodictis do form Spardochium. Some form very large colonies which can be visible with the naked eye. But microscopy is frustrating because the spores are very dark and you can't always see the septum position inside of them because they're too dark. And this group has not been revised by specialists yet, so most of the names are obscure and used more or less arbitrary. So this is a group that needs some love. Microscopium is another one that probably needs some love because it have yet to be sequenced, yet it is very common actually. And it produced rather big, slender, dark cinemas. However, the canidias that protrude from the top a bit like an Olympic flame in some sort are quite pale. The canidia look a bit like Fusarium's canidia, but they are somewhat pigmented and less of a boat shape usually. The best way to find Morphographium is to find the big bark chunk from a dead fallen trees that are not really decayed yet, especially trees like elms or maple. If you look at this with a hand lens, you probably will find a few eventually, although they are rather solitaries and with rarely more than 2 or 3 in the same area. They can be confused with pin lichens as well, which look quite similar to this, honestly. Paradidymobothrium, which is also another long name, look a lot like Morphographium, but can easily be separated by the fact that its canidia only have 1 septum in them. I've only found it a few times on wood, and Tetelomorph is a group of disc fungus that you can find really often on wood, which is called Molesia. Molesia are little bluish discs found on rotten wood, but the Molesia themselves are very difficult to identify, unfortunately. But this anamorphic state is distinctive, in my opinion. I should probably talk a few minutes, shortly about the penicilliums, which are, like Aspergillus, very variable and numerous in numbers of species. With okay, never mind. Never mind, sorry. I got a message and I thought I was going too much off time or something. I'll be done in 5-10 minutes, I promise. So it is very variable and some are even cinematis, such as Penicillium glandicola, which as its name may hint, grow on old acorn. There's also a cinematus one that found on droppings of animals, but I've never seen that one. There's some interesting Apomycetes growing on droppings, but you have to be a bit courageous to look there. So Penicillium can be found on pretty much any kind of habitat, and they're famous for the production of several toxins, including probably the most famous of them all, which is called penicillin, which is used as an antibiotic. Pseudoargyridia cordicalis is more or less considered a bulbil, but not quite. It produces oddly branched chain of angular-shaped Canadia that almost look like shirts of some sort. I can't really describe the shape. They are often found with other similar species, such as Candelabrum. However, Pseudoargyridia tend to be bluish, while Candelabrum tend to be yellowish, and both are pretty under the microscope. Pseudoargyridia also imply that we also have a real Argyridia around, and we in fact do, and the Argyridia candida form quite large bulbils on rotten woods. The Pseudoargyridia is best found on very rotten wood in extremely wet habitat near marshland, and in those habitats it is common. Spadicoides are also common on wood around there, and I know there are several species, but I've only found 2 of them. And the main difference between those 2 is that one have 1 septate Canadia and one have Canadia with no septate. So Canadia form on very small pegs, as you can see on the lower right. Sporidesmiums are pretty much always Nottyakis fungi, and like Monodictus, more or less of a wastebasket group, although there have been a book on this group, fortunately, which solved their relationships somewhat. Most have very long Canadias with many septum. I think some have up to 20 or 30 septum, if not more. And yes, the number of septum can be useful for identification, and also the length of the Canadia and how the Canadiaphore look like. They're common on wood and bark, but they can be found on various habitats. Arthrosporium lignatil superficially look very similar to the Bactridium I talked earlier by forming yellow cushion on wood. But if you look under the microscope, you will see that the spores are completely different. Those spores are huge for Canadia by being more than 50 micron in width or so, and almost completely globose or circular shape, and filled with an unidentified yellow content. Those Canadia are in chain, and it is relatively common, and I'm working on this group right now. It is quite an interesting fungi too, I think, which showed quite a lot of mystery, I think, especially on regard of why you need to evolve giant Canadia to begin with, when so many form small Canadia and do fine. Stilbellas are probably one of the, probably the most obvious group of cinematas, the classic cinematas, a beautiful lollipop, if you can say. Some are reds, many are white, some are yellow. They can be found in various habitats. Many are tropical. In fact, the red one were found temperature bearing stuff from the Carleton University butterfly show greenhouse. So even the greenhouse, I guess the greenhouse is in Ottawa, so I guess it counts as an Ottawa find. The microscopy can be difficult, but it is often needed for identification. And the color of the Canadia is also very useful. Like the red ones are rather distinctive. Tilak Lady and Brachiatum produce very indeterminate cinematas that look like almost like a bottle brush or a toilet brush of some sort. They are rather specific in their habitat, and they grow on old rotten mushrooms, as you can see on the upper right. Well, there was an old rotten mushroom just next to the regular one, but it's all decayed now. The Phylites, this species produce Phylites even if it's not really obvious. And the Phylites are quite long, reaching several, maybe 30, 40 micron in length, as you can see on the lower right. The scale is in microns, so from 0 to 20 microns. I probably have to talk about Trichoderma a little bit because it is probably the most frustrating group of fungi around. Personally, it is extremely common on rotten wood, and it is extremely voracious in culture, and it will contaminate your culture whenever getting to occasion and ruining your chance of growing rarer stuff. But furthermore, they also need they almost always need to be looked under the microscope to be identified, and all of the microscopic elements are small. The telomorphic state is hypocrea, which produces a yellow cushion in the lower right with little spots. And the yellow is a nice example of the telomorph and the anamorph growing together. There is one exception for Trichoderma, which makes this genus awesome, to my opinion, and it is Trichoderma flavipes, which look nothing like the other Trichodermas, but genetics said so. So I guess that's how it is. It produces an all yellow cinema usually, or sometimes grayish. And if you look closely at the hairs that surround the stalk, you can see that those hairs are all spiky, which is quite diagnostic for this species. It looked a lot like a stilbella, and it was called stilbella until a few years ago. I don't see it very often, but I found it in a wide range of habitat, including wood. Verticilladium is not necessarily my most interesting species, but I want to feature an important point in mycology, and it is that sometimes pictures are just not enough for showing what you want to show. For example, on my picture on the top, you may think that those structures are Phylites because they look in a similar branching. However, if you look very closely with the microscope, you can see that the condyles actually form on little scars or pegs from the Phylites. I was not able to take a picture of this, so sometimes a drawing is really the best option and shouldn't be discarded. Drawing is valuable in mycology, especially for small Hyphomycetes. You can find this one, if you want to find this one, you can find it on rotten needles of conifers on the ground. And I want to conclude with probably my true favorite fungi, because I might very well be the first one to see this, and it have no name yet it is completely unique and completely different. It produces a mass of beautiful cinematas joined by the base on a common substrate that look like a cushion. The Canadia break off the conidiophore a bit like bullets, and I interpreted this as somewhat of atalic reproduction, but it's not really. Furthermore, if you look closely on the upper right and upper left, you will see that the stem is all spiky, while on the lower left, you can see all those big cells surrounding the stem. All of those traits are unique to this fungi, which I do not know what its relative are at this point. And I need to find it more often, but I've only known it from this single spot in Orleans. Hopefully someone here will find it, and if so, please let me know. I find this one really fascinating. The only time of the year I find it is in mid-June to very early July. By mid-July, they're all decayed and all old, so it seemed to have a very specific season, unlike most of the other ones. And I want to finish with 2 pages of some books that may interest people, although unfortunately most of those books are kind of rare, if impossible to get, and if you can get them, probably quite expensive. So although they might be available in specialized libraries, the most essential of all of those, if you're really interested in Hyphomycetes, and probably the easiest to get, would be the Genera of Hyphomycete, which was made mostly with the contribution of Keith Seifert, which may really well be still in this room right now. He's watching this talk right now. So, and he's the leading expert in Hyphomycetes, and he's amazing. So, this book is really a must-have if you're interested in Hyphomycetes in general. Although it is perhaps not the best to identify two species, it will usually help you narrow down to a specific group, which make it easier to look on later. Other interesting books include Dematiaceous Hyphomycetes and more Dematiaceous Hyphomycetes, which are made specifically for Hyphomycetes with dark Canidia or dark conidiophores. So if you have one of those, these books are your best bet. There's also a book called Microfungi on Land Plant and Microfungi Miscellaneous Substrates, which are quite interesting because they show a huge variety of fungi, but not only Hyphomycetes, but all the microfungi that you may find on a specific species of plant. If you're interested in Simulomycetes, there are also some good books for those, which I suspect are very hard and expensive to find, unfortunately. Probably the best one to get would be the Colomycetes Fungi Imperfecti with Pycnidia, Acervilla, and Stromata, or Colomycetes Anamorph with appendage bearing Canidia. Other interesting books with lots of illustrations, although they're a bit old and maybe a bit outdated in their names, would be the genera of fungi sporulating in pure culture, which include many Hyphomycetes, although this only includes species that have been known to grow in culture, of course. And illustrated genera of imperfect fungi, which is a bit like the genera of Hyphomycetes I just said, but much older. And there are also a few books on specific group. For example, there's a monograph which describes as many species of Eliquospores fungi possible. It is very old, but the illustrations are beautiful. It's honestly a work of art. I won't lie. There's also another book by Keith Seifert, which is a monograph of Stilbella and some allied of Hyphomycetes, which focus on those big cinematus Stilbella that I discussed earlier. And there's also another book, which is focusing on wood inhabiting molds of New York. There's not many pictures in this one, but it is a good list of fungi of molds that you might encounter locally, and may be a good starting point, especially with another book such as the genera of Hyphomycetes. So this is it for me, and I think I will just stop sharing my screen. And if you have any question, well, you can put them into chat if you want, or probably raise your hand. I think you can. Apparently, you're able to raise your hand even if it's virtually. So thank you very much for listening. It was very appreciated, and I hope you liked it. It's weird without the applause, isn't it?
Good clap.
Thank you very much, Johnathan. Are you there, Ian?
Yeah, I'm here. I'm still having network problems.
Oh, it's not
Okay.
you.
All right, did you saw at least a few things? Or... I have big name in here.
Are there any questions? I'm trying to figure out where is the raising the hand button.
Well, I think people can right now. They can probably just unmute and ask any questions if they need to.
Okay, good.
I have a question.
Yes.
Johnathan, the one that you found that's unique,
Yes.
what kind of wood was it growing on?
On really rotten poplar wood in flooded forest in spring. So
Okay.
One after it dried out.
Okay. That's a beautiful.
It is.
It's
I
beautiful.
can't believe that. I don't think there's a name for it.
Yeah. Well, so you have to come up with the name.
Yeah, I'm trying to, but I have issues with growing it into laboratory, which would help me a lot. So I'm keeping trying. I'm pretty close to have enough information for naming it. I do have a working name, but I'd like to describe it further one day.
How big is it?
It's those cushion are about 5 millimeters, maybe in width or so. So it is pretty small, unfortunately. It looked like a Ceratio mixta fructulosa a bit.
Yep.
I had to convince a few people that it was not that when I first showed them to.
So we all need to be looking for it.
Well, if you want to.
Yep, we'll find it for you.
Yeah, it's really pretty. So I like it a lot, and like I say, I like it even more because I don't think it's named yet. So
I have another question, but I think there's some on the side.
Some in the chat, some questions.
yeah. Oh yeah. Joey asked me if I look at foam. I only looked. Well, Joey's one of my former colleague, and he's quite specialized in Mollesia and things like that. So he's quite an amazing microscopist too. But I think I don't really look at foam that often. I look because I was curious about it, and I indeed found a few of it aquatic Canadia. As I've said before, all of those pictures except for the life cycle at the start are all by me, and they're all from local areas. So yes, I did look at water foam a few times, but not very often. Even if I would like to, probably. It's quite fun.
It's Lynn here.
Hi, Lynn.
Were you able to find all of those things you showed us with your hand lens? Were you able to see them in the field with your hand lens?
Many of them, the Spardochium and the Cinemas for sure, but some of the others not easy. Some of the others not easily. Others you will see a big mass of them, like it look almost like a very small crust fungus growing on the wood, or a discoloration of the wood, which is actually the mass of Asexual Canadia spore. But some of them you really need a microscope, a dissecting microscope to see, unfortunately. Really a hidden world, so.
Johnathan, if I can ask another question.
Go ahead, I'm not in a rush.
You said that things are changing, that anamorph and the telomorphic have often different names for the same fungi. And it sounds like from what you said that that's changing going forward, that they should have the same name. But what about the ones that already have names that are different? Or is that changing? Do you know? Yes,
they will. One of the name will have to be removed. So already a few of the most familiar fungi, of some of the more familiar fungi have changed names. So now in taxonomy, maybe you're familiar with Hypochrea, which produce a yellowish cushion with green spots on the wood.
Yep.
This name, which is the anamorphic name, was not as much used as the Trichoderma name. So right now we are calling them as Trichoderma in some websites, and I think it's pretty much official now. So Hypochrea have been scrapped, and some of the fungi that, like the one that grew on truffle, I think, like Ophiocordyceps, which you may have heard of or seen, have changed for its anamorphic state, and it's now called the Tulipocladium, because the Tulipocladium was better known than Ophiocordyceps in the literature. So, so usually it's the reverse. Usually it's the anamorphic name that gets scrapped, and people, those names are often like 100-year-olds, and just now some of those relationships are being realized due to more advanced techniques such as sequencing, to realize that, hey, those two things are actually the same ones all along. So then we have to change the name. So this will probably happen to Armameys soon enough.
What is the word mold synonymous with hyphomycete? Are they
I would,
synonyms?
I would say no, but maybe I'm biased. I think that mold, it is on a large extent, but I think mold is more arbitrarily used to any microscopic fungi that are inconvenience to us. So I think what you call bread mold are not always hyphomycetes. So you can also be a psychomycetes, which are not even closely related. But I think molds tend to have a more negative value to them. So I prefer to stick to hyphomycetes. Because it's kind of hard to call those beautiful stilbella molds, I think. Especially when the first thing you see mold, you see that moldy bread that probably have Penicillium or Trichoderma growing on it.
Jonathan, Keith has a question that if there was one hyphomycete that you think mushroomers could find and would find interesting, what would it be?
Most of them are pretty small, unfortunately, but I think that the last one I showed with no name is probably something that some mycologists would find interesting. Actually, there is one that I could think of, which is called Dendrocolibia. I will write the name right here. Dendrocolibia race. We'll write the name into chat. Just give me one instant. Dendrocolibia racemosa, which is a hybrid between a gill mushroom with the anamorphic state producing cinemas on the stalk. Lynn know this one. I've showed a picture a few weeks ago. This one would be very interesting on the East Coast. I think every mycologist would be able to find it. And on the East Coast, there is only a handful of record of this species. It is apparently super rare, and it is absolutely beautiful. And there have been a very credible record of it near Ottawa a few months ago. And now I need to find it there. I want a good picture of this. This is probably one that come in mind of the big ones that could interest any mycologist to look at least in my opinion, but I can't really show a picture right now. But I think you should Google Dendrocolibia racemosa. And I think Keith would probably agree that this one is striking, although he might not like the name right now. As this one also had, this one is a good example for the one name two mushroom rule, isn't it? The cinemas was named after the asexual fungi, while the gill mushroom that was the sexual state was named differently. So now only one name need to stand. And it's the mushroom name that seemed to be prioritized right now. But yeah, if you find that one, let me know too. There's only 3 records of it in Eastern North America that I know of. While it is pretty common on the West Coast and Europe, and somewhat common in Europe, so. But I think that other fungi would be interesting, like the one that I talk, and Bactridium is also interesting, Spardochium, Stilbellas in general, although all of these are pretty small. So that's it for that question. That's a good question, actually. And Neriman is a specialist in molds too. So in hyphomycetes, she focused on Fusarium, Penicillium, and Aspergillus. So she's quite amazing too.
Now you said, can I ask another one?
Sure.
Another quick question from Joan. You said that you were studying one of them right now in the lab. What does that entail?
Well, studying them in the lab is usually growing them in in petri dishes to get their growth characteristics, and to perhaps more importantly nowadays, even if some people may disagree, including me to some extent, to get sequences. So nowadays it seems that most of the best way to publish a mushroom is to get sequence and to prove that it's not the same as something else. So I have quite a few that I'm studying, and I have a few that are on the verge of publication too. So this is quite exciting, honestly.
And when you say to sequence, you mean DNA sequencing?
Yes, extracting the DNA and sequencing. We do not do whole genome sequencing. We only sequence a specific part of the DNA that tend to be just variable enough to distinguish between fungi, but not variable so that we can still classify them easily. We try usually with a few of those regions. So.
Is it difficult to establish the relationship between the anamorph and the teleomorph?
Depends. You can establish it rather easily if you're lucky with sequence. Sometimes it's frankly impossible because some teleomorphs do not produce anamorphs. And also, frustratingly enough, some anamorphs, despite being asexual, do not apparently form a teleomorph, or if so, do not normally form them. So, but in some cases it is possible. Some cases it is the best to just get the spores from the teleomorph in a petri dish and wait for them to grow. If you're lucky, you will get the candida of the anamorph in there. If not, sequencing is a good option. Or if not, it can be more challenging though. It is quite interesting, especially if it's not a relationship that you would suspect, and you have to prove it with several sequences usually. Or especially if you're with
I think that would be fun.
Yeah, it is. Especially if you have an anamorph that look different from the anamorph in that group, and that's kind of annoying, or in a group that don't have any known anamorph or vice versa. So it's kind of interesting, honestly, but sometimes it's not always possible to prove.
Any further questions? Okay, well, I guess that's it. Johnathan, thank you very much.
Thank you for the invitation. I think I did okay. So.
That was fantastic.
Thank you, Jonathan.
Very interesting. Thank you.
Thanks, Joe. Thanks to Joey too.